Hello, everybody. So I guess that was the end of the introduction video. So thank you all for joining us today for this edition of NannoTalks Volume 2. Today, we're really happy to welcome Samuel Phelps, who's a postdoctoral researcher at Harvard University working on isotopic fractionation in coccolithophores. And so without further ado, I'll let Sam go ahead and share his screen and his research with you all. Thanks a lot.
Awesome. Thanks very much, Clara, for that introduction. It's a pleasure to be zooming in today to speak with you all about my research. Today I'll be talking about Noelaerhabdaceae coccolithophores as a proxy for past atmospheric CO2 concentrations. And I'll build this up through calibration to application and explore what it means for Neogene climate. Let's see, there we are. So before I dive in, I just want to give a few shout outs to my funding agencies and collaborators. This work is a summary of my PhD research. I'm lucky to have some wonderful collaborators in Heather Stoll, Claire Bolton, and Luke Beaufort. I did my PhD at Columbia University with Prodigal Polisar. And I'll show some work from culture experiments that I was fortunate to work on with Gwen Hennon in Sonia Diamond's lab at Lamont Doherty Earth Observatory. So just for context, we know from direct measurements of the atmosphere and from measurements of gas bubbles trapped in polar ice that the current atmospheric CO2 level is higher than at any time in the last 1 million years or so. When we look at projections for the rest of the century, estimates range between about 400 and 1,000 parts per million by 2100, depending on how aggressive and successful emissions reductions and carbon capture efforts are. But what does this mean for the future? And so one of the questions motivating my research is this: just how much will temperature increase and how much will climate change given the projected change in atmospheric CO2? Better understanding the relationship between CO2 and temperature, particularly in past warm intervals in Earth history, with CO2 similar to today and to those levels projected for the future, can help refine these estimates. So how far back in time do we have to go to get to modern CO2 levels? And what about for CO2 levels projected for the future? People have been working on this problem for decades, and there's a rich literature of reconstructions that paint a broad picture of the evolution of CO2 over the Phanerozoic. And it generally corresponds with our understanding of climate over the Cenozoic. Higher CO2 levels are found in the early Cenozoic, which accompanies global warmth, such as here in the Eocene, where there are palm trees in the Arctic. And the CO2 declined and global temperatures cooled, ice built up in the Antarctica and later in the Northern Hemisphere. But one time period that stands out in particular is the Miocene, between about 23 and 5 million years ago, so this period here. And we know from several lines of evidence that it was a time of intense global warmth with substantial ice retreat. And in Antarctica, sea surface temperatures were about 10 degrees C warmer in the high latitudes. There were forests in places that are currently permafrost. And much of our knowledge of this time period comes from the alkenone CO2 proxy, which is one of the what I will be talking about today. And so just kind of from a first principle standpoint, the algal CO2 proxy, or paleobarometer, is based on observations of carbon isotope fractionation in plants, where CO2 comes into the cell through diffusion and is converted into organic matter by rubisco. And carbon made from photosynthesis has the potential to record environmental information because rubisco, this enzyme shown schematically here, this enzyme converts CO2 into organic matter. And so through this carboxylation reaction, it prefers carbon-12 to carbon-13 by about 11 to 30 per mil, or 1 to 3%. And I'll use this delta notation here and this epsilon notation throughout, where delta is the ratio of the 12C to 13C in a substance relative to some standard. And epsilon is basically the difference between delta values of two substances. So in this case, oops, apologies there. In this case, it's the difference between the organic carbon and the CO2 from which it was made. So shown here is a cartoon schematic demonstrating the underlying theoretical principle. When CO2 is plentiful, up in this top panel, the kinetic preference for rubisco is more strongly expressed, and the organic matter has more of the light isotope. This gives it a more negative delta 13C value because it contains more 12C. And the epsilon P value, a term I'll be using a lot, is the difference between the organic carbon and the CO2 from which it was made. And that value is larger when CO2 is more abundant. And when CO2 is less abundant, the CO2 concentration around Rubisco will be lower, and this kinetic preference can't be fully expressed, resulting in organic matter with relatively less of the light isotope, relatively more of the heavy isotope, giving it a more positive delta 13Z value and a smaller EP value. And so the classical method for interpreting alkenone or algal epsilon p values is based on diffusive CO2 supply, where CO2 diffuses into the cell and is converted to organic carbon. In this model, the relationship between epsilon p and CO2 can be described by a line like this, with higher EP values at high CO2 and lower EP values at low CO2. And so this 25 per mil value up here has traditionally been taken to be the maximum expression of fractionation by Rubisco at the limit of infinite CO2. So basically, if there is no limit on the amount of CO2 around the cell and the cell can be as selective as it likes, the maximum Rubisco fractionation that you can get is about 25 per mil. But because of physiological differences between algae, different species have different relationships and may not fall in the same line. And I'll talk more about this complexity later and what causes these deviations later on. But so early on, efforts were made to target molecules that have a unique or monospecific source. And so what we end up using are molecular biomarkers called alkenones, which are produced by the coccolithophorid algae of the family Noelaerhabdaceae. And these organisms are special because they make these long chain ketones that can be easily analyzed in sediment samples. So we'll measure the carbon isotope ratio of these molecules, specifically the 37,2 methyl ketone, which is shown here on top. It has these 2 double bonds and a ketone head group and 37 carbons. And we use the carbon isotope ratios of foraminifera to estimate the delta 13C ratio of the inorganic carbon from which these alkenones are synthesized. And we calculate EP roughly as the difference between the two. And there's a rich body of literature studying alkenone carbon isotope fractionation through time. Here I'm showing published records of alkenone EP over the last 45 million years or so. They document a general decline in EP from around 25 or 23 per mil in the Eocene to about 12 per mil in the Pleistocene. And below I'm showing the benthic foraminifera isotope stack with upwards on this plot indicating warmer or less glaciated conditions, and down on this plot indicating colder or more glaciated conditions. And this EP record generally corresponds with climate cooling over the Cenozoic, so the higher EP in the hothouse of the Eocene. But we see an increase in EP as climate is cooling in the Miocene. And so if you were to interpret this record just in terms of CO2, as the conventional model suggests, then we would actually interpret this as an increase in CO2 over this time period, which does not necessarily agree with our understanding of climate through this era. And when we look at the Pleistocene, where CO2 changes are known, the first record to try this suggested that glacial to interglacial changes in CO2 could be reconstructed from alkenone EP, which was very exciting. This is the pioneering study by Jasper and Hayes in 1990, showing a close correspondence between the ice core CO2 record and the CO2 levels estimated from alkenone carbon isotope ratios. But as more data were generated over the last 20 years or so, it became clear that not all locations behave the same way, and that alkenone fractionation is likely more complicated than the classical or conventional method assumes. And so this is perhaps unsurprising when you consider the physiology of the coccolithophores that are producing these alkenones. So the traditional model of understanding carbon isotope fractionation of these organisms is based on land plants. You could think of this as a simple box where CO2 diffuses in, CO2 is converted to organic carbon, and it's just a simple passive diffusive model. But in reality, Rubisco has a slow turnover rate and a low affinity to CO2. And when CO2 is not abundant enough, it can actually act as an oxygenase and oxidize organic matter and generate CO2. So the concentration of CO2 in the surface ocean today is generally below the half saturation constant of the enzyme, suggesting that ambient CO2 levels are suboptimal for CO2 to behave as a carboxylase. So to overcome these limitations, the chloroplast has a low permeability to CO2 to keep CO2 concentrations high in the vicinity of Rubisco. And algae also take advantage of the abundant DIC, or bicarbonate, in seawater, so they can actively take bicarbonate into the cell, pump it across the cell membrane into the chloroplast, convert it to CO2 through either enzymatic or acid-base equilibria, and then use the CO2 generated from that conversion for photosynthesis. And we see experimental evidence for this activity. On the right, I'm showing some culture experiments of Noelaerhabdaceae show an increase in the expression of genes used for anion uptake and carbonic anhydrase as DIC and CO2 decline. And on the left, I'm showing experiments that measure the uptake of CO2 and bicarbonate in Huxley, showing a shift to less CO2 uptake and more bicarbonate uptake as CO2 availability decreases. And so taking these together, it suggests that the kind of traditional interpretation of diffusive supply only might be too simplistic. There's another interesting piece of information that builds on this, and in this model, the maximum fractionation is 25 per mil, where at infinite CO2, Rubisco should be fully expressed. And if you measure Rubisco from a spinach leaf or from a tobacco leaf, in vitro, you can measure a value of 25 per mil. And some early experiments projected an intercept up to 25 per mil, and so that made sense. But in recent years, the Rubisco fractionation from Noelaerhabdaceae was measured in vitro, and the value returned from that was 11 per mil. So it requires a different explanation to achieve EP values greater than 11 per mil if the actual kinetic fractionation by the enzyme is only 11 per mil. And in addition to non-diffusive carbon uptake and these intricacies with the kinetic fractionation factor, some experiments have shown a direct influence of irradiance, or the amount of light energy on carbon isotope fractionation, where both higher light intensity and longer photoperiods lead to higher EP values. And so all of this is to say that the classical interpretation of alkenone EP in terms of CO2 alone might be too simplistic. And so what I'm going to walk you through next is a series of studies that have tried to improve our understanding of alkenone carbon isotope fractionation and bring some life back to the alkenone paleobarometer. And to do this, I'm going to start in cultures where we can calibrate the proxy and probe some of the fundamental principles. Then we'll move into the ocean and look at core top samples to see how the calibration transfers out of the laboratory and into the natural environment. Then I'll aim to validate this method by comparing EP variations in the Pleistocene to the known influence of CO2 and cell size. And finally, I will apply this method to a sediment sequence from the Neogene to glean new insights about CO2 change, climate change, and the carbon cycle over the last 20 million years or so. So first, let's look at the calibration. And the driving questions here are what factors control carbon isotope fractionation in alkenone producing algae? And does the traditional diffusive model describe carbon isotope fractionation? So let's think for a second about what can modify the expression of fractionation by Rubisco and the CO2 signal in algal carbon isotope ratios. So decades of work have shown that it's not just the CO2 supply that affects EP, but the carbon demand of the cell relative to the CO2 supply. And so it's really the balance of these two processes that affects the CO2 concentration inside the cell, how much of that CO2 is consumed and converted to organic matter, and how strongly the fractionation by Rubisco is expressed. So carbon demand is proportional to the growth rate times the carbon content of the cell, so how much carbon is it fixing. Larger cells and faster growing cells have a higher carbon demand. The surface area over the cell sets the area over which CO2 can diffuse into the cell. So that sets the permeability, the CO2 supply, and the surface area set the diffusive CO2 supply. And these cells are nearly spherical, so we can measure the cell diameter and approximate them with a spherical geometry. And in the diffusive model, the maximum EP would occur at 25 per mil, and we would predict that these variables would affect EP in these directions. So what we're going to do is look at culture experiments where these parameters are tightly controlled. And what I'm showing here is a compilation of culture studies over the last 15 years or so, along with some new data from our study where we grew Gyphera oceanica in batch culture. This was recently published in G Cubed a few months ago. And so what we find is that epsilon P does indeed decrease with increasing carbon demand relative to supply. So this decline here is what we'd expect kind of from first principles. As the carbon demand increases relative to the CO2 supply, the carbon isotope fractionation declines. But we also see that the shape of this is not linear as prescribed or as expected from the diffusive model. And so this black line is shown here, and we see a big departure from the diffusive model as carbon demand relative to supply increases. And what we also see is a clustering of warm colors at the top of this plot. And the symbols here are colored by the total irradiance that the culture receives in a day. So in moles of photons per meter squared per day. And we see that warmer colors populate the top part of this plot, so high EP values, and cooler colors populate the bottom of this plot. And if we look at this in a different direction, so here it's the same data, but with daily irradiance on the x-axis and colored by the carbon demand relative to supply. So if we look at this relationship in more detail, what we see is that high irradiance and low demand relative to supply are required for high epsilon P values. So earlier I showed EP change from about 23 per mil in the Eocene to about 13 per mil in the Pleistocene. And it's possible that almost all of that could be explained by irradiance changes. And so that would be changing from about 23 per mil up here, high irradiance, to about 13 per mil low irradiance conditions. So it seems that irradiance is really driving a lot of the variability in culture experiments, and that it's a parameter that we really do need to consider when we try to interpret sedimentary epsilon P data. And so what we do here is we make a simple empirical model of the factors influencing EP and see what it takes to explain all the variance in epsilon P in the culture experiments. And this follows the approach of a study led by Heather Stoll in 2019. That was really the pioneering breakthrough in this. And so what we do here is, let's start with the left side. Here we're just including the parameters in the conventional diffusive model. So that's the cell size, CO2, and growth rate. And when we include those variables, we can explain about less than 50% of the variance in epsilon P. And when we include irradiance components here on the right, so that's the light hours and the light intensity, we can explain almost 90% of the variance in EP in this culture data set. An important thing to note here is that growth rate is actually, at least in this model, the way it's parameterized, growth rate does not appear to have a significant influence in the data. And so the p-value here is actually about 0.5, so it's not statistically significant. So that means that moving forward, as we look into the environment, we will not have to quantitatively constrain growth rate using this model. But so just to summarize, the behavior of EP that we find in the culture experiments when we put these data together and just look at the variables that are driving the variance in EP, it's different from what the traditional models have suggested, and that the kind of a diffusive supply framework is not sufficient to explain the variance of EP that we see in culture experiments where everything is tightly controlled. And so this is really just an extension of the work led by Heather Stoll in 2019. And so these predictors are slightly different from the model in that study. But we'll take this next out of the laboratory and into the natural environment. And so the driving question here is, do these same patterns observed in culture quantitatively describe alkenone EP in the modern ocean? And so the things that we need to consider are irradiance, CO2, and cell size. So fortunately, we have a way to constrain cell size. Some really fundamental work by Dierenje Hendricks found intact coccospheres in sediments throughout the Cenozoic and measured the cell diameter and the coccolith length and by SEM. And that study in 2007 found that coccolith length and cell diameter are very highly correlated, and that's shown here in this right panel. This has been confirmed in culture experiments. There's some data by McClellan et al. 2016 that has a rich data set of culture experiments that come to the same conclusion. But so what this means is by measuring coccolith length in sediment samples using microscopy, we can then estimate the geometry of the cell and constrain cell radius as one of the inputs into the EP model. So for this study, what we did was we produced new core top EP and coccolith size data. We compiled existing core top and water column particulate data. And for this presentation, I'll focus on the core tops because what we're really interested in is reconstructing CO2 from sedimentary alkenone and EP. The particulate results are a little more complicated and nuanced, mostly because the environmental conditions at the collection depth at which the water was pumped at a single point has a Lagrangian history because the alkenones are produced over the course of days, but the sampling condition only represents one depth and as a point sample in time. But what we also did here was we compiled all coccolithophore assemblage and size data that you find in the literature. And we examined the influence of irradiance, size, and CO2 on alkenone EP. And in the core tops, the approach here is empirical, but what we do is we set the depth of production to 75% of the mixed layer depth. We calculate the irradiance at that depth and use that as the input in the model. And when we do this, what we find is that we actually have a reasonable ability to model alkenone EP in core top sediments. And so the relationship here is scattered, but using the multilinear regression model derived from the culture experiments, we're capturing the magnitude and direction of EP change in core top sediments. And so some of the scatter in the left panel is related to uncertainty in the measurements and some from the applied irradiance value. But when we look at the residuals shown on the right, so it's the culture calibration, the residuals of the calibration in the gold color and the residuals of the modeled versus measured in the sediments, they share the two curves share a lot of structure, and so some of the error in this measured versus model plot is related to the structural uncertainty in the calibration. But so what the main takeaway from this for me is that we are actually able to quantitatively take these transfer functions developed in the laboratory to the natural environment. And because we can model EP with some degree of accuracy and precision, we can then extract environmental information from it going down core. So I want to take a quick detour and talk about one of the other findings of this study. It's currently in revision at G-Cubed and will hopefully be out in the next few weeks or month. But so earlier I showed how EP is proportional to cellular carbon demand relative to diffusive CO2 supply. We found support for this relationship in cultures, although the relationship is not linear as prescribed by the diffusive model. So the conventional model applies a linear relationship between EP and demand over supply, shown here, where lower EP occurs at higher growth rates, and because these are spherical cells, at larger cell sizes as well. So in paleo applications of this conventional method, because cell size and growth rate were difficult to constrain, all biological factors other than CO2 were lumped into this B parameter, which if you're familiar with the literature, if you've seen some of the works upon which this is built, this equation might be familiar to you, where this EP equals 25 minus B over CO2. And so 25 is that the intercept of this plot, which is the maximum fractionation by Rubisco. So at infinite CO2, the limit of this is 0, and the EP value would be 25 per mil. And the B value is essentially the slope of this plot. So early calibration work showed that the B value that you needed, if you went out to the ocean and you took a sample from the surface ocean and you measured the CO2 value, you measured the epsilon P value, and plugged them into this equation and solved it. Early work showed that the B value that you needed to solve that equation was highly correlated with the phosphate concentration in the seawater in those samples. So what I'm showing here in this left panel is the canonical work by Bidigari et al. In '97 that calibrated the alkenone CO2 proxy by sampling suspended particulate matter in the ocean. And at the time, this was an amazing finding because it provided a way forward to constrain B in the natural environment. If you know something about the trophic status of the location, if you know the phosphate concentration and can assume that it's relatively constant through time, you could then use this calibration, estimate a B value, and interpret EP values in terms of CO2. And so this was a major breakthrough at the time. And because cell size in modern alkenone producers was thought to vary little between these sites, the idea was that B mostly represents growth rate. And the correlation between B and phosphate was considered a correlation between growth rate and phosphate. And so this relationship, or a slightly modified version of it, has really been the basis for all alkenone CO2 reconstructions over the last 20 years, until just about the last year or two, where things have really started to change a bit. But so over that time period, much more modern data was collected. And so when I compiled all of the available data, the relationship between B and phosphate is actually much less robust than the original calibration work. And so shown here on the left panel is the same relationship as the previous slide. The black symbols are the same data from Bidigari et al. In '97, and shown in the blue symbols are all the data that have been generated since 1997. And it appears that the relationship between B and phosphate arises because of an underlying relationship between phosphate and CO2. And so this left panel is B regressed against phosphate. This right panel is CO2 regressed against phosphate, and the R-squared values are almost identical. And so what this is telling me is that the B is not actually telling you anything more about carbon isotope fractionation than the CO2 concentration. And additionally, there have been abundant field campaigns that actually measured growth rates of alkenone producers using in situ labeling experiments. So isotope labeling experiments where you put an incubation data, or you collect some seawater in a big cubitainer, spike it with an isotope label, put it back at depth and incubate it for a day, and then measure how much of that label was taken up, and you could calculate the growth rate from that. So if we actually look at measured growth rates in the field down here in this bottom plot, regressed against B values from the same sets of samples, we find that there's no relationship between B and growth rate as expected from a diffusive model. And so this is more evidence that the diffusive framework just might not describe how these organisms fractionate carbon either in the lab or in nature. So in lieu of a robust mechanistic understanding about carbon uptake and intracellular carbon isotope conversion, because we are still working on unraveling that mystery, we can instead use an empirical model like the culture-based multilinear regression. And so I just showed previously how it's the core tops samples suggest that when IRradiance is treated consistently across sites, we can model EP with reasonable variability, or with reasonable accuracy, but that's IRradiance at various locations today. And what we really want to do is model at one point through time. So because we don't yet have a robust proxy for past irradiance changes, we'll have to assume that it is constant through time or allow it to follow some envelope of uncertainty. So our ultimate goal is to invert the model and solve for CO2 from alkenone EP. And to do that, we need to identify places where irradiance variations are small enough that EP and size are telling you about CO2. And so that takes us to the next section. So where can alkenone EP be modeled through time using known CO2 changes and constant irradiance? And if irradiance is not stable in the Pleistocene, we really have no faith in it being stable in deeper time. And so if we can show that irradiance is relatively stable over the last 800,000 years in some sediment samples, that provides us confidence for going deeper in time. And so to do this, we generated 2 new records of alkenone EP and coccolithophores, compiled and standardized existing alkenone EP records so they could be interpreted together. And we compare modeled to measured EP in all sites where size is known. And so here I'm plotting out locations where alkenone EP and coccolithophores are measured. And at each location, EP is modeled using known variations in CO2 from the ice core record, along with measured coccolithophores to estimate cell size changes. Because we don't have a bona fide proxy for irradiance, we have to assume that it is constant, and we treat it as constant in all records. So a slope of 1 on this plot suggests that the measured EP is responding properly to CO2 and cell size changes, and the irradiance variations are small. And an intercept, this is the 1-to-1 line in this faint dashed blue line. On the y-axis is the modeled EP, on the x-axis is the measured EP. And so a slope of 1 is what we're looking for good performance. So we see that there are 2 locations that really stand out, ODP 807 from this study and ODP 668. And 925 also goes in the right direction from this Zhang et al. 2019 study. There's been some recent work by Alba Gonzalez Lanchas et al. That just came out over the last couple of months that also found a good performance at site 925. And I'd encourage you to check that study out. But what we see is that these 2 sites seem to pass the first test of good performance in the Pleistocene with constant irradiance. So this analysis serves as an exercise to help identify which locations are promising for deeper time work. Of course, irradiance does not have to be stable on multi-million year timescales, but we view it as a prerequisite for attempting to reconstruct CO2 change. And let's see, yes, this is the study I just mentioned by Alba Gonzalez Lanchas, and who worked at ODP 925 over MIS 12-9, and found that the culture-based model, a slightly different version, can also model EP changes well from the ice core CO2 variations. And so we're finding agreement with this updated approach. And so what is it about locations that appear to behave well? The irradiance at depth is a function of the irradiance at the surface, the attenuation coefficient, and the depth of production. And so when we look at the modern monthly variability in each of these parameters, the irradiance at the surface, the attenuation coefficient, and the mixed layer depth, what I'm plotting here is the monthly standard, or the standard deviation of the monthly average values of each of these parameters at these locations on the x-axis, and the slope of the modeled EP versus the measured EP at each of these sites. So perhaps unsurprisingly, sites with oceanographic conditions of low variability, so kind of low latitude, deep mixed layer, minimal dynamics perform best. So that's where a slope is close to 1. Those locations all have a low variability in the attenuation coefficient, the surface irradiance, and the mixed layer depth. And so to go deeper in time, we'll target sites that have low variability in these parameters, and that's where we'll head next on this journey. So let's step in deeper into the Neogene and use this updated alkenone paleobarometer to ask how atmospheric CO2 has changed over the last 20 million years and see what this means for our understanding of climate, biosphere, and carbon cycle changes. So the late Miocene brought about the expansion of C4 grasslands, plants that have a competitive advantage in low CO2, but there's still, we also saw dramatic global cooling, enhanced at high latitudes, a reduction in marine carbonate burial, but there's still some uncertainty surrounding the direction and magnitude of atmospheric CO2 change during this time period. So what we did is we generated data from a site in the Western Equatorial Pacific that was identified in the previous section as a site with good performance in the Pleistocene, where we could model EP reasonably well with ice core CO2 variation and measured cell size changes. And so in this framework, EP is a function of CO2, cell size, and irradiance. So to get the irradiance value to use down core, we calibrate the Pleistocene samples by solving for irradiance with the known CO2, the measured cell radius, and the measured EP value, and apply a constant irradiance value down core. And when we do this, we see that CO2 declined by about 600 ppm from the early Miocene to the Pleistocene. So these are the data shown here. The epsilon P record has a slight M-like structure with a maxima in the middle Miocene. We found that coccolith length and inferred cell radii were up to about 2 times higher in the early Miocene compared to the Pleistocene. And all else equal, given this empirical function, larger cells require higher CO2 values to produce the same EP value. So what we see is about a 600 ppm decline in CO2 from the early Miocene to the Pleistocene. And these results find support from independent work at ODP site 1088 by Thomas Tanner and Heather Stoll's group. And so we're starting to converge on this idea that Miocene CO2 is actually perhaps quite a bit higher than previously estimated. So let's look at what this CO2 history means for global climate and environmental change. The bottom panel shows our new CO2 estimates on top of the benthic delta HNO stack. And I took the averages of the middle to early Miocene, late Miocene, and Pliocene. And the top panel is showing temperature anomalies from Herbert et al. 2016 broken into different latitude bands. Turning SST into global average temperature is difficult, but these red boxes show estimates for the late Miocene and the middle early Miocene from global paleoecological data. And of course, these reconstructions have their uncertainties, but the late Miocene was likely 4 to 5 degrees C warmer than pre-industrial. The middle Miocene was maybe 7 to 8 degrees C warmer than pre-industrial. And before any quantitative analysis, we can see that these higher temperatures are associated with higher CO2. What I'm showing in the middle panel is the timing of the initiation and expansion of C4 grasslands around the globe. So this open circle is the first evidence of C4 in the landscape, and the closed circle is when they dominated the landscape in each of these regions. And we see that the global expansion really seems to occur after about 9 million years, when there's a big stepwise decrease in CO2. When we look at the data quantitatively in terms of Earth system sensitivity, which is the equilibrium temperature response to a doubling of CO2, we see that values fall between about 4 and 6 degrees C for a doubling of CO2, which is in agreement with estimates from the Pliocene and the Eocene. So this is an exciting result, I think, that shows that CO2 decline in the Miocene accompanied global cooling and grassland expansion. I'm going to shift gears a little bit into the final section of this and just show you one final finding from this work. And so the two main proxies we have for the carbonate system are the algal CO2 proxy and the boron isotope pH proxy. These figures I'm showing here from the Research Coordination Network. It's this NSF sponsored project to kind of vet CO2 estimates and compile them in a publicly available database. And so the website for that is this paleoco2.org website. I'd encourage you to check it out. There's a lot of great resources there, and all of the CO2 data that is available basically has been compiled there and vetted. But if we think back to basic marine chemistry courses, there are kind of 6 components of the marine dissolved inorganic carbon system: CO2, bicarbonate, carbonate ion, DIC, alkalinity, and pH. And with 2 of these 6 parameters, you can calculate the rest. And so that's what I'm going to do here. I'm going to take these new CO2 estimates, pair them with existing estimates of pH from the boron isotope proxy, and solve the carbonate system. I think in the, let's see, actually, just for a little background, boron isotope ratios in marine carbonates are really a pH proxy. Like the carbonate system, the speciation of borate ion and boric acid is pH dependent. As pH increases, more of the total boron exists as borate ion. And so these 2 chemical species have an offset of about 27 per mil. And because of isotope mass balance, the delta 11B value of borate ion increases, pH increases. And marine carbonates record the delta 11B of borate and the signal of that pH. So that's how we can measure boron isotope ratios and get back to a pH value. Here's a beautiful flow chart from the PaleoCO2 website that I described that explores all the parameters that go into these calculations. This is more detail than is necessary right now, but the key thing that I want to point out is I'm basically just focusing on these last 2 steps. So second carbonate system parameter is going to be the CO2 aqueous record that we generated in previous slides and the existing pH records. So if we look at existing pH data for this time period, this is the record from Sosdin et al. 2018 with the axis inverted. So more acidic is positive here, more basic is down on this plot. There's very little change in pH through the late Miocene and into the Pleistocene. It's slightly more acidic in the Miocene. And so in this study, these authors converted pH estimates to CO2 estimates by estimating surface DIC concentrations from benthic delta 11B pH records, assuming a deep ocean carbonate ion concentration based on the depth of the lysocline, or sorry, the depth of the carbonate concentration depth, and a secular change in calcium ion, and an assumed constant DIC change from surface to deep. And so instead of that, we could take the CO2 record and pair it with this pH record, and then calculate the DIC that we get from this. And so that is shown here in the bottom panel in purple. And so what we find is that DIC, given this higher CO2 and relatively basic pH, the DIC needed to be quite a bit higher in the Miocene, up to about 50% to 100% higher than in the Pleistocene. And alkalinity, given the relationships of the carbonate system, was also likely higher in the Miocene compared to today. So this is definitely a different finding from many of the inferences either modeled or inferred from secular variations in calcium concentration and in and using the calcium carbonate compensation depth as a proxy for carbonate burial. But when we look at the sedentary record, do we see any support for this finding in the literature? There's some early work by Baptiste Citrus-Marx and Jurinja Hendricks in 2014 that was built upon by some work by C. and Rosenthal in 2019, suggests that carbonate burial in the Miocene was much higher than it is in the Pliocene and Pleistocene. So in this study, about 2 to 4 times higher than in the Pleistocene. And these authors here argued that higher burial fluxes were maintained by higher weathering fluxes and greater alkalinity supply to the ocean during the Miocene. And so what could support these higher alkalinity fluxes? And perhaps the answer lies in volcanic outgassing, which is an external lever on the slow carbon cycle. So global compilation of rift length, shown in panels A and B here, shows more extensive geological rifting in the Neogene compared to the Pleistocene, suggesting that CO2 outgassing was higher, driving higher CO2 levels, global warmth, and enhanced weathering in the Miocene. So this is all still a bit of a work in progress. We're working on expanding these findings to other sites and kind of digging into this more quantitatively. But I think the existing data suggests taking both the pH data and the CO2 data at face value, which we have no reason not to, the solution is a very different picture of the carbonate system than has been previously put forward. So just to conclude, what I've shown is that alkenone EP records CO2 variations, but not through the diffusive model. And IRradiance, as others have demonstrated before, is a key independent influence on EP. EP can be modeled in core top sediments, suggesting the same factors that play in ocean, in the ocean. For paleo reconstructions, minimizing variability in IRradiance is essential, and actually developing a proxy for IRradiance will be a major contribution in my view. And we find a large CO2 decline in the Neogene consistent with the expansion of seafloor grasslands and global cooling, and pairing this with boron isotope pH records suggests that alkalinity and DIC were much higher in the Miocene compared to today. So thank you very much. With that, I'm happy to take any questions.
Okay, so if anybody who has a question who's in the room can raise their hand, then feel free, go ahead. So thanks, Sam, for a great talk. Super
My pleasure, thanks.
Okay, while we're waiting for people to wake up, I have a question. My question's about IRradiance and how you could potentially look at changes in IRradiance in the past. I mean, obviously it's good to choose sites where you think that there's been little change in IRradiance, but how realistic do you think that really is? And is there some way, by looking at past insolation or orbital parameters, we could try and model changes in mixed layer depth that might be proportional to changes in PAR at production depth?
Definitely, yes. That is the question that keeps me up at night. I think the insulation variability is relatively small on that we can account for, maybe plus or minus 10% or something. It's really changes in the production depth, as you mentioned, and the mixed layer depth are really the kicker, because given the exponential attenuation in IRradiance, let me just go to that quickly. In many locations, if you change IRradiance by, let's show this one. I just want to share this again. So here's a hypothetical IRradiance at depth curve given two different attenuation coefficients and different surface IRradiance values. You could have a relatively small change in the production depth, let's say from 50 to 25 meters, that could actually change IRradiance by a factor of 2. And so that is, so really trying to constrain the depth of production is the major question. I think there are some promising both micropaleontological and geochemical proxies that might get after this. And so there's some work by Ivan Hernandez Almeida that looked at the difference between deeper dwelling and surface dwelling foraminifera in the oxygen isotope difference between the two. Can tell you about the structure of the mixed layer. I mean, Clara, you have work looking at this as well for a different, to a different end, but same means, to kind of generally characterize the trophic status and the structure of the upper water column. I think really throwing all qualitative and quantitative indicators we have at it is going to be necessary. So that's kind of like coccolith flouristics, also to look at whether there are indicators of more eutrophic or oligotrophic status.
I guess the difficult bit then becomes making it quantitative.
Yes, absolutely. There's some culture work that suggests the hydrogen isotope ratios and the coccolith carbon isotope ratios tell you something quantitative about irradiance, but those calibrations are definitely in their infancy. So it's an active area of research for our group and others, I think.
Thanks. There's a couple of hands that have gone up in the room. So Mario, would you like to go first?
Hi all. Thank you, Samuel. Well, there's a lot to digest in your model, so I'm not able to understand it all. But if I understood correctly, your data suggests, your last data suggests that there might be some changes on the quantification of CO2, of PCO2 during the Miocene related to the previous interpretation. So how does it change? Do you have a graph that shows you where your values are positioned in terms of the forecast, the present day values of PCO2 and the forecast for the next for next year? Do you have let's say an update of the first record or the first graph that you showed us?
Yes, I unfortunately do not. That is a good way to characterize it, but the maximum estimates that we're getting in the middle Miocene are around 800 parts per million. And so that if we go back to that first figure, we're somewhere up here between 600 and 800. So in line with the business as usual or slightly reduced business as usual. And yes, there's Clara's 2016 paper, Bolton et al. 2016 in Nature Communications, and the Stoll 2019 Quaternary Science Review, and Tanner et al. 2020 in Paleo are all records that have kind of, or all studies that have put forward an updated alkenone or updated CO2 record for this time period. And so there's some literature out there that if you're curious, I would point you to.
Okay, thank you. Thank you very much.
Yeah. Thank you.
Thanks, Mario. Ines, would you like to ask Tam your question?
Yes, hello, Samuel. And I would like to ask you, did you find out in your sample some diatoms?
I was not looking for diatoms in these samples. Most of so at least in the like Pleistocene Neogene sequences that I am working on, they're 90% carbonate for the most part. So biogenic silica is not a major component of the lithology there.
Okay, because I'm asking, diatoms are excellent proxy for paleo environment that you try to study with boron, but diatoms are much better to use for alkalinity and other things, nutrients, proxy, and those things that you also incorporated in your study. And it's much better for those results are diatoms.
Okay, that is, thank you.
Because they are very sensitive environmental parameter, and they will tell you some diatoms are good for organic matter. They said it's very high organic matter. You have very high alkalinity, low alkalinity environment, eutrophic, oligotrophic. They are very good really for environmental proxies. As environmental proxies, sorry.
No, thank you. That is wonderful to know. I have not looked at the silicious component of these at all, really, or in detail. But I will definitely keep that in mind, and finding sites that might have a larger breadth of potential proxies that we could exploit would be very helpful. So if there are diatoms that are good indicators of alkalinity change, that I would be very, very interested in that.
Oh, yes, they are good, and they can say about something or about circulation, upwelling system, and those things as well, not just
Sure.
oligotrophic alkalinity, temperature. salinity. For those things, they're very good. Thank you.
Thank you. Thank you.
Thanks, Ines. I see Patrizia has her hand up.
Yes. So thanks a lot for the presentation. It was actually really nice, and I have many questions, of course, because it was quite complex in some parts. But first of all, I was a bit surprised about some of the results that you show. And first, like when you compare the culture calibration with the sediments, I was surprised in a way to see this very well a correlation with the probability model that you show, because of course the culture is very limited. And you were mentioning first about the size, that you don't expect to be like a very large distribution in size in the normal sediments, but I mean, depends what you mean for that. So that was my question.
Yes, I might have been a little confusing and conflated things there. So we, when I was mentioning that the limited size variability, that was the assumption that went into the kind of like conventional Bidigary calibration. So the assumption in that work was that size didn't vary much in the modern environment, so that you could basically treat it as consistent throughout that calibration. But in our samples, we did measure size, and it ranges from between about like 2 microns to 3.5 microns or something like that. So we do have constraints on size, and it is quite variable in the modern ocean. So the application of the culture-based model to the sediment samples includes measurements of size. So we measure the size, as well as we estimate the CO2 from kind of the GLODAP database, with an accounting for the anthropogenic influence also. And so we use those, and then the irradiance at 3/4 of the mixed layer depth in each location to calculate EP. So there is substantial size variation there, but when we include that quantitatively in the model, we still are able to kind of get much of the same structure.
Now I understand.
Okay.
Thanks.
Yeah,
the second
thank
question was about the irradiance, and I saw the site that you work on is an equatorial site. So the deep chlorophyll maximum is, so the production layer, so the signal that eventually you have in the sediment is about 100 meters or more. So most of the production is quite deep in this site. So I'm wondering, like, so when you are reconstructing the CO2 based on this site, that is a very deep production layer, what are the implications in terms of like CO2 reconstructions that are not at the interface with the atmosphere?
Yeah. So that's a great question. The mixed layer at this location is quite deep. It's about 100 meters, I believe. So it should still have some. It's not. You're right. It's not at the interface, but it should still have good communication with the atmosphere. And so it's not a place where there's really substantial outgassing, or there is some vertical upwelling today. It was likely a little bit below the equator around 10 million years ago. But yes, that's something that we are struggling with also. I don't think it's not that the production today is not deep enough. I don't think that you're really getting a respiration signal of CO2. And given its kind of relatively stable oceanography over the last 20 million years, and it hasn't moved that much, it didn't go like from a gyre to a upwelling site, that I think the majority of the signal that we would be seeing is something that is static and consistent. And so even if it is a bit of an elevated CO2 signal relative to the surface and the atmosphere, I would argue that it's likely just offset in that direction. And we actually calibrate it to the Pleistocene, so we're accounting for that already when we go down core, because the irradiance value is just that we apply is just whatever irradiance we need to get the Pleistocene to work, basically. So it is, if the actual CO2 value is a little bit higher, I think we are correcting for that. I would be, or I'm eager to apply this to other modifications through this time period, and there's some data that I can do that with that I just haven't been able to pull together for this yet. But I think once we can combine several sites and look at the general trends, some of those uncertainties we should be able to eliminate. I hope, but we'll see. But thank you for the questions. I appreciate it.
Thank you.
Okay, for those of you that are still there, I had a couple of announcements I wanted to make. The first was that if you're planning on coming to INA 18 in Avignon next year, there's a little expression of interest form that we'd love you to fill in to help with our planning. I'm going to put the link to that in the chat. And secondly, our next NannoTalks 2 speaker is going to be Blanca Alcin from the University of Salamanca, exactly 1 month from now on November the 18th. So I'll just give you that link, and thank you very much for joining us. It was great to have such a big turnout at NannoTalks, and thanks Sam again for such an interesting talk.
Thanks so much for having me. Thank you all for listening and for your questions. Appreciate it.
Okay, that's the link in there for the INA 18 expression of interest for those of you that might not have seen it on the Facebook page, etc. Thanks everybody. Have a great rest of day.
Thank you.